Okay, this is the video lesson over Unit 6, Eukaryotic Organelles. You need to make sure you are following along with your guided notes and completing them as you watch the video. Please pause and re-watch any parts that you need to relearn or re-watch that you didn't quite get the first time. Pay attention, make sure you take complete notes because you will need this information for the remainder of the week's activities. A quick recap of our cell theory before we go into any more about cells. We have three parts of the cell theory. Remember, all living things are made of cells. Cells are the basic unit of structure and function in an organism. And lastly, all cells come from the reproduction of existing cells. I know we've already talked about this, so this is just a refresher recap. The cell membrane model. All cells are starting up from the outside moving in. If we start at the outside, all cells are going to have the cell membrane. Cell membrane is going to be composed of phospholipids and proteins that are able to move bilaterally. That word bilaterally means that it's able to move from side to side, short distances. We're going to spend all of Unit 7 going into lots of detail about the cell membrane structure and how things are transported across the membrane. The membrane actually makes a certain kind of pattern, and because of the proteins and different parts of the membrane, we call this pattern the fluid mosaic model, and you need to make sure that you recognize that as a name for the cell membrane. Next, we're going to talk about the control center of the cell, and this would be our nucleus. The nucleus is not always in the center of the cell, but it is concentrated in the cell, concentrated region that has its own membrane called the nuclear membrane, and we have a different name for this. We can also refer to this as the nuclear envelope. So you need to be familiar with both terms when I'm talking about the membrane surrounding the nucleus. As I said before, this nucleus serves as a control center of the cell. So if we were in our school, what do you think would be the control center of our school? Think about what part of the school that would be. We're going to be going on a tour later this week where we are going to figure out how the parts of the school are like the parts of a cell. We have nuclear pores, and these are little holes you can see down here on the picture that are going to allow things to enter and leave the nucleus. Now, they are guarded. Okay, not everything can get in the nucleus. It's a highly restricted area. So only certain molecules are going to be allowed in or out of the nucleus. Right? DNA never leaves the nucleus. If you look here in the middle of the cell, you can see this region here, which is the blue. That is the nucleolus. And the nucleolus's job is to make ribosomes, which will be important for the cell's ability to produce protein. Remember, the DNA lives inside the nucleus and it never leaves, making the nucleus our control center of our cell. DNA and actually has two different forms. It can either be in the form of a chromosome. Okay, if you look down here, you can see the chromosomes look way more organized. Or that is only going to be in dividing cells. Or it can be in the form of chromatin. And I think of chromatin kind of like a bowl of spaghetti. Okay, you can see over here it's not very organized. It's kind of all just mixed together. And that's what it's going to look like in non-dividing cells. So when we talk about cell division next 9 weeks, we will use the words chromosomes and chromatin quite frequently. Next, we're going to discuss the cytoskeleton. The cytoskeleton is made of proteins called microtubules, microfilaments, and intermediate fibers. Okay, the cytoskeleton is like the beams inside of a building. It's going to hold the cell up and give it its shape and structure. So it provides support for the cell and helps it to also move because it's going to allow the cell to constrict or expand and able to kind of like wiggle its way along through its environment. Again, this provides the structure and support for the cell and helps it to keep its shape. So it keeps its shape. You might want to add that down. That's important. Lysosomes. When I think of lysosomes, this prefix lyso makes me remember Lysol. And we use Lysol to get rid of things that we don't necessarily like or want around, like germs and things like that can make us sick. Lysosomes do that for the cell. This is a sac containing digestive enzymes that are going to break down cell waste, such as old cell parts and pathogens. A pathogen being like a bacteria that infects the cell, or it could be a virus. something that gets in the cell that's not supposed to be there. It's the lysosome's job to take it out. Now, the lysosome can also play a role in what we call apoptosis, and this is programmed cell death. This is basically when there's something tragically wrong inside the cell, and the cell needs to die so that the rest of the organism does not become affected. This is cell suicide for the good of the organism. This could be like a rogue cell whose DNA has a mutation in it, and if this cell continues to grow and replicate, it could develop into cancer. So apoptosis is one of the safeguards that we have, that our body naturally has to prevent bad cells from living in our body. The vacuole, this is usually the easiest one to remember. So the vacuole is our storage space. It stores water, food, and waste. You're going to notice that in plant cells, it's going to be huge. It's going to take up a lot of space because plant cells need to store a lot of water in order to do photosynthesis, but not necessarily in animal cells. So an animal cell is going to be quite smaller because we don't need to store things as much as plants do. And it's not going to be present in bacteria cells, okay? In fact, none of these organelles are going to be present in bacteria cells. Centrioles. These are made of microtubules, which you might remember from the cytoskeleton slide. Centrioles kind of look like, they almost look like little tater tots, is what I had a student tell me one time. And they are only seen in animal cells during cell division. Okay, they're going to be helping to pull apart the chromosomes, the DNA, in cell division to make sure that each new cell gets the correct amount of DNA. So we'll talk a lot more about centrioles when we talk about cell division next 9 weeks. Cilia and flagella are the means of a cell being able to move. These are also made of microtubules, and they're arranged in a 9+3 arrangement. That means we have 9 on the outside and then 3 in the middle. And it's arranged in a circle, right? And they help with the cell to be able to move. Cilia, we've talked about cilia. They are the small whisker-like fibers that beat back and forth. So they're like really small hairs on the edge of the cell. So if you look down here at the bottom of the screen, you can see these little green fibers coming off the cell. On the left, those would be cilia. They allow the cell to detect changes in the surroundings and also help the cell to move. Flagella are these long whip-like tails that come out from the cell. Okay, these are going to whip back and forth and allow the cell to move. In humans and animals, we have only one type of cell that has a flagella, and that is a sperm cell. Okay, it allows that sperm cell to move where it needs to move. Okay, so the function of the flagella is to move the cell forward. Ribosomes. Ribosomes are one of the most important. Organelles, and they can be found in 2 different places. They can be found free-floating in the cytosol, or that's also called the cytoplasm, or they could be attached to the surface of the rough ER. Okay, and we'll talk about the rough ER in a little bit. The ribosome is made of proteins and RNA. Remember, these ribosomes are produced by the nucleolus, which is in the nucleus. And its function is to produce protein. And remember, all cells, regardless of whether you're a prokaryote or a eukaryote, has ribosomes, because all cells have to be able to produce protein. Ribosomes have a large subunit and a small subunit, and we'll talk more about ribosomes and their structure when we discuss protein synthesis. A little bit more detail about the cell membrane or the plasma membrane. We've already said that it's made of phospholipids and proteins. These phospholipids look like this. We have a polar head and non-polar tails. Okay, so when they are connected, they're going to arrange with the polar heads facing out, okay, and the tails facing in. So it looks kind of like this, right? And that's because of the hydrophobic and versus hydrophilic. The middle here is going to be hydrophobic. The outside will be hydrophilic, okay? Don't worry too much about trying to figure out the structure of the cell membrane, because that is all what we're doing in Unit 7. And on the cell membrane, we also have proteins. They can either be on the outside or inside, or they can be stuck into the membrane, right? Depending on what type they are, it's going to determine whether they are peripheral or integral. This is an important thing here, okay? This is a function of our cell membrane, right? It is selectively permeable, meaning it is picky and chooses who it's going to allow into the cell and who it's going to keep out. Okay, so you can kind of think of this as like a guard at the door of the cell. It's going to decide who's good to come in and who is a threat and isn't going to be allowed in the cell. So our cell membrane is our boundary that's basically going to determine who gets access to the cell and who's denied. Now, in addition to the cell membrane, I want to discuss the cytoplasm here. The cytoplasm is the gel-like material plus all of the organelles that exist between the nucleus and the cell membrane. So the cytoplasm is anything that's not an organelle, basically. We also have things called glycoproteins that can be on the cell membrane. These are proteins that have a carbohydrate attached. These are important in helping the cells to recognize each other. So all of your cells in your body have certain glycoproteins, which is kind of like a name tag. Okay, it's kind of like a name tag, and your cells use this to recognize that, hey, you belong here, or hey, you don't belong here. We need to alert the immune system. Cell membranes also contain the steroid cholesterol, right? And this helps to keep our fluidity of our membrane so that we're not becoming too rigid, because we do not want a rigid cell membrane. That is not good. All right, the mitochondria is our powerhouse of the cell. It's surrounded by a double membrane, and we know that it contains its own DNA because we've discussed this when we talked about the endosymbiotic theory. The role of the mitochondria is to burn sugars to make energy for the cell. Okay, energy in the cell is called ATP, right? And we'll talk more about that when we talk about cellular respiration. Okay, so you can look over here at the mitochondria picture I have, and you can see the folded inner membrane here allows for more surface area so we can have more reactions happening at the same time. This is a huge advantage because we can burn more sugar and get more energy at a faster than if we just had a single layer. Okay, because it's folded, we get a higher surface area. Again, this is our powerhouse. This is what does, you need to write this down, it does cellular respiration. This is what does cellular respiration. Not the breathing of the cell, but breaking down the sugars to get energy. That's why it's called the powerhouse. Right, our Golgi apparatus, or Golgi body, sometimes it's referred to, is kind of like the UPS of the cell. Okay, it looks like a stack of flattened pancakes, and you can see down here, this is our receiving end. As the molecules move through here, they're repackaged and they're shipped out to go out of the cell. Okay, so the function is to modify, sort, and package substances from the ER for transport out of the cell. So that's why I call this like UPS. It takes in the stuff from the ER, it packages it, it boxes it all nice, puts a nice little bow on it, and then it ships it out to its final destination. And here's where we talk about the endoplasmic reticulum. This is simply a network of internal membranes, and we have two different types. The first type is the rough ER, and we call it rough because it has ribosomes attached to the surface. So if you look over here on the right, the blue one that I have that you can see, that is the rough ER. You can see the ribosomes give it this bumpy texture, okay, because it's covered in ribosomes. And its job is to make the protein in the ribosomes, and then the ER actually modifies the protein to its final shape, and then exports it to the Golgi apparatus, where the Golgi will determine its final destination. The smooth ER does not have ribosomes. That's why it's called smooth. So the function of the smooth ER is different. You can see it over here in the orange color. It's a lot smoother. It's even shaped a little bit different. And its job is to produce lipids, and it's also going to help in detoxifying the cell, making sure that there's not any harmful chemicals inside the cell. So we're going to see in liver cells, for instance, liver cells help to detoxify our bloodstream. So our liver cells are going to have a higher concentration of smooth ER as compared to other types of cells in our body. A chloroplast. Now this is one organelle that us animals do not have. Okay, a chloroplast, like a mitochondria, is surrounded by a double membrane. It also has its own DNA. Okay, it has an outer membrane that surrounds, but we have separate inner membrane, just like in the mitochondria. Okay, the inner membrane sacs are called thylakoids, and this is what contains that green pigment called chlorophyll that's going to capture the sunlight to do photosynthesis. Right? So this is only going to be found in plant cells and some protists like algae that are going to be doing photosynthesis. I'm not going to expect you to know the parts of the chloroplast quite yet, but we will learn those when we do photosynthesis. All right, cell wall. This is another part that we do not have in our cells as animals, because cell walls are going to provide protection and support. All right, it's a rigid structure, meaning it's not going to allow for a lot of movement. And so we're going to find these in plant cells, right? And remember, in plant cell walls, we talked about during biomolecule unit, that they contain the carbohydrate, the structural polysaccharide, called cellulose, which is going to help make those plant cells nice and sturdy and give the plant its structure. Now, we just finished talking about prokaryotes in Unit 5, and remember that prokaryotes also have cell walls, but in their cell walls, they're made of peptidoglycan instead of cellulose. Remember, this is not in animals, only in plants, fungi, protists, and bacteria cells. Right, so that was eukaryotic organelles. We just talked about prokaryotes. So in contrast to eukaryotes, remember, prokaryotes do not have a nucleus. They do contain a single strand or plasmid of DNA. Okay, but like the cells, like eukaryotes, they also are surrounded by a cell membrane, and they also contain cytoplasm. Right? So this concludes the video over all of the eukaryotic organelles. You need to make sure that your guided notes are complete. Make sure you are familiar with the function of each of the organelles. Okay, you will be having a quiz over this at some point, so I expect you to be fully prepared. When you are done, you need to make sure, we need to put these in our journal at some point so that you do not lose them. All right, that is it, and I will see you tomorrow.